this the real life? Is this just fantasy? Caught in a landslide, no escape from reality. Open your eyes, look up to the skies and see.
And see chromosomes, genes, and alleles. DNA profiling is a huge breakthrough in forensic science. And if we go back to what you remember, hopefully, about biology, the nuclear DNA, the genes that make up a person, are found in the nucleus of the cell. Now, they don't have this chromosome shape most of the time, only when the cell is preparing to divide. Usually it's more of a spaghetti type of chromatin form, a little bit more messy. But if we take these chromosomes, which condense upon the preparation of the cell to divide, and we unwind them, what we find is that those little strands of DNA are curled up around themselves, and we unwind that, and we find that they're wrapped around histone proteins, and then eventually we get down to a single layer called the double helix. Now the double helix has two strands of deoxyribonucleic acid, and those two strands are united in the middle, basically where the rung of a ladder that you would step on, by hydrogen bonds. This is an electron microscope image of a chromosome. Okay, so in terms of the chromosomes that you personally have, and every person has, as a human being, you have 23 pairs, totaling 46 chromosomes. Now, one of each pair you got from mom, and one of each pair you got from dad. And in a karyotype, those chromosomes that you got from mom and that one from dad that match up, in other words, they code for the same genes, even if they don't say the same thing about those traits, they code for the same traits, they line up in the form of a karyotype. Now, you can have mom's genes say one thing about a trait and dad's genes say another thing. And when that happens, and we have, let's say, an allele for blue eyes and an allele for brown eyes, we call that heterozygous. When mom and dad's alleles, which an allele is a form of a gene, when those two alleles are the same from mom and from dad, then we call that homozygous. And if it is a trait that is dominant, we call it homozygous dominant. And if the two are the same and they are both recessive, we call that homozygous recessive. Okay, so let's look at what that plays out to in forensics. This is known as DNA fingerprinting. Of course, fingerprinting is figurative here. Fingerprinting in the sense of actual fingerprints goes back years in forensic science history. But DNA fingerprinting is also known as profiling, and so a fingerprint being unique to that person, but this time instead of it being ridges on the actual finger appendage, it consists of bands of particular DNA sequences. And the idea is that this is a type of individual evidence that nobody on the planet, except for maybe identical twins, has the same band pattern. This is just another look at what we talked about with the double helix, and you can see the base pairing that happens in the middle, and remember this is united by hydrogen bonds here. We call these nitrogenous bases, the pairs of the molecules that make up the rungs of the ladder. And if you remember back, we have adenine, cytosine, guanine, and thymine. Do you remember which pairs with which? If you said that A pairs with T and C pairs with G, then you would be correct, and only that base pairing will fit together. So, in what's evaluated with DNA identification, generally polymorphisms are the places where evaluation takes place, and these are non-coded DNA segments that have repeated base sequences. So poly means many, many forms. So there's segments of DNA that are not coding for a trait that we know of, but in some people there are certain numbers of copies of that particular segment, and in other people, maybe there are fewer copies of that segment. And so if we look at multiple polymorphisms across that person, then that combination of the different numbers of those sequences can be the part of individual evidence. Eventually, through a technique that we're going to talk about in a moment, the DNA fingerprint appears as a pattern of bands of those polymorphisms that are separated out on X-ray film. So there are two types of polymorphisms, or repeating DNA sequences. The first is variable number of tandem repeats, and this can vary from 9 to 80 bases in length. The second is short tandem repeats, which is just 2 to 5 bases in length. And when I say bases, I mean A's, T's, C's, and G's. So think about it. Would you rather write something 2 to 5 times or 9 to 80 times? Well, if you can have the same effectiveness for both, you'd rather do the shorter one. And so short tandem repeats are more of the go-to, and then VNTRs, variable number, would be if increased clarification or something else is needed there. And this can be used for tissue matching as well as inheritance matching. So if two samples have the same band pattern, then we can assume they're from the same person. Let's say that there was a blood spatter found at the crime scene, and even though red blood cells do not have a nucleus, therefore no nuclear DNA, white blood cells in the bloodstream do contain that nuclear DNA. So we can evaluate the DNA fingerprint from those white blood cells. We can also evaluate a known sample of DNA, for example a cheek swab or something like that, from a suspect, and if they're the same, we assume they came from the same person. In inheritance matching, this is a way of figuring out biological parenthood. We can evaluate the bands, and in this example, the mother's on one side, alleged father's on the other, child's always in the middle. If a child has a band that the father doesn't, well, that doesn't necessarily mean it's not the dad. We have to say, oh, okay, the mother had that band, so she could have provided that for the child. And over here, the mother didn't have the band, so the father does, he could have provided that. Over here, the child does not have the band, father does, but the mother does not, so the child could have inherited the lack of the band from the mother. Okay, so in this last example, mother does have it, child doesn't, father doesn't. I see no problem here saying that this father is still possible of being the biological father. Now over here, child has a band, dad doesn't, but that's okay, mom does. This is a problem. Child has a band, didn't get it from mom, didn't get it from dad, and so our results here is that paternity is not established in this case. Zooming out a little bit, population databases are helpful in understanding probabilities with DNA sequencing. So we have to understand the gene variations that occur among individuals. If we know the probability of a certain allele being present in the population, then we could determine the probability of these allele combinations in this DNA sequence, suggesting that it is the person whose blood was left at the scene. And so if there is a low probability, that's not very convincing evidence. If it is an astronomical number in terms of 1 in 9 million, let's say, of a chance that this would be a random person with the same alternative form, that's a pretty convincing form of evidence. Now, places where DNA can come from. We mentioned blood, also saliva, because cheek cells are deposited in that saliva, skin, hair, seminal fluid. This is a form of individual evidence, can identify a specific person, can also be classified as trace evidence if it's in small amounts, and polymerase chain reaction is a technique that generates multiple copies of DNA evidence. So let's talk first about how you collect it, then we'll talk about how you analyze it. So, as most forms of evidence, you want to use gloves, you want to avoid contamination, you want to air dry it and put it in a paper evidence bag, because a plastic bag will hold in that moisture, potentially grow mold, and contaminate the evidence. Okay, so preparing the DNA for fingerprinting. We're going to mix the DNA with some enzymes, which are going to cut apart the DNA at specific fragments that you want to evaluate. And then you're going to separate it within the agarose gel to make those band patterns. And you do that by passing an electric current through it. So this is big time here. We're going to talk about DNA profiling. Step 1 of 4 is extraction. This means we're extracting the DNA from the cell to be able to analyze it. So whichever type of cell that it was a part of, we're going to disrupt that cell and then extract it from the nucleus and be able to get just a concentrated sample of just that DNA and get the cellular debris out of the way. You may have actually done something like this in biology class with strawberries or bananas, extracting the DNA from there. Second step is amplification. And this is what we mean by polymerase chain reaction. This is an important step because if you have just a small DNA sample, then that doesn't leave much room for error. You need to have copies of what you're about to do so you can run multiple tests, or if something goes wrong, then you have a backup. So we've talked about VNTR analyses. PCR, or basically amplifying or copying that, can be used for that, as well as the STRs. Okay, here's an example. Here's our template DNA, and the red part represents the wanted gene to be able to analyze. And the first cycle of amplification, we end up getting 2 copies here, and then each of those copies will form copies, and it becomes exponential, and you now have lots and lots of copies of DNA to work with, and that's called amplification. Third step is electrophoresis, where we actually run the DNA samples through an electric current in an agarose gel, kind of like a jello consistency. So you're going to inject the DNA into little crevices or wells in the gel, and then due to electricity flowing through here, you're going to make that negatively charged DNA travel from a negative side to a positive side, right? Because opposites attract. So you're going to have negatively charged DNA due to its chemical composition of phosphate groups on the outside of the DNA. And so that DNA will then travel across the gel. Now, I played point guard in college, and I was not the biggest or the tallest out there, but man, I was fast, right? And so if you can think about a 6'5" individual, which I had in my team, not very fast, versus a 5'4", but 5'6" in the program, individual next to her, then that 5'4" individual is going to make it further, right? Smaller, less friction. And so the shorter the band of that particular DNA segment, the further they're going to make it through the gel. And that constitutes a staggered band pattern. So here's an example. And we might say, okay, if the crime scene sample was sample 6 and my suspect is sample 7, not a match. Oh, and notice that the base pairs go down in descending order. So 100 base pairs is the shortest segment and made it the furthest along, whereas 500 base pairs, longest segment of A's, T's, C's, G's, didn't make it as far. Sorry, 6'5" teammate, who will remain unnamed. DNA probes are just a way to track particular segments of DNA. What can be done is to attach a fluorescent tag to a segment of DNA, and when that part is amplified, then that particular DNA fluoresces, and so you can literally track that fluorescent tag through the analysis. And in most criminal cases, 6 to 8 probes are used to be able to track that DNA. Okay, so we'll take a look at the bands and widths being significant in matching the samples of DNA. Yellow represents the child could have gotten the band from either parent. Okay, a child got the band from mom in this case, inherited no band from mom in this case, inherited no band from dad in this case, and then inherited this band from father. And as far as we know, this child could be the biological child of both mother and father. Okay, analysis of DNA fingerprints. DNA fingerprinting can do the following. Crime scene DNA with a suspect match, determine maternity, paternity, or another relative. Eliminate a suspect, free a falsely imprisoned individual, which has done many times in the case of the Innocence Project, which has gone back and used DNA evidence that didn't exist at the time of that case, and in some cases led to exoneration for wrongful conviction, as well as identifying human remains. Alright, thanks for tuning in. Read the summary, enjoy the outro, take care.